Hi, everyone. I'm Tracy. I'm in the invertebrate lab, and we're happy to host our speaker, Dr. Charles Boch. And I was given this introduction to read. Charles grew up along the coast of Southern California, where he failed to earn a living surfing and fishing. After exploring alternative options, he went on to earn his PhD in marine science from the University of California, Santa Barbara, where he studied the effects of seawater temperature and light dynamics on coral spawning synchrony, as well as developing cost-effective restoration methods. After finishing graduate school, he became a researcher at the McKelly Laboratory at Hopkins Marine Station, investigating the effects of seawater temperature, dissolved oxygen, and pH on kelp forest communities. Currently, Charles conducts field and laboratory studies with Jim Barry at MBARI and multi-institutional collaborators looking at the effects of ocean acidification, ocean warming, and hypoxia on the biology of abalone, including fertilization, larval development, juvenile survival, and growth to inform population models and management. So today, he has his talk up there, but he'll be presenting Oro del Mar, Chasing Abalone Life History Under a Changing Climate.
Thank you very much for that introduction and for the invitation to speak here at Moss Landing. It's a pleasure to be here. I know we're neighbors, but it's always fun to come over here and say, oh, you guys have a beautiful place too. So I'm going to tell you a little bit about the work that I've been doing with the collaborators at Hopkins and what we've been doing at MBARI for the past year and a half, but the work actually goes back further than that. Oro del Mar is actually a euphemism given to abalone because oro is gold of the sea. Commercial fishermen used to say, it was back in the heyday in the 50s and 60s, they used to say it was like picking up $100 bills off the ground. Think about that. And by the end of the day, they were several $1,000s richer. Those days are long gone. You can see some of the work that we've been doing on upwelling.stanford.edu with all the collaborators. It's interdisciplinary work, collaboration between physical oceanographers, modelers, experimental biologists, ecologists, and social scientists as well. So when we think of the climate, especially for the Pacific Basin, we generally think of the Enso Index. And we see a graph like this. And what do we generally think when we see the Enso Index? Hot years, cold years, really, really hot years, really, really cold years. And for global predictions, we have an estimate about 2 to 3 degrees in our lifetime or in 2 lifetimes. So, but what about other covariant environmental variables? There's a prediction that it's going to decrease 0.4 pH units, and there's some evidence of expansion of the oxidant minimum zone. So we take this and we go, what does this mean for the abalone or critters that live in the local areas that are living in the coastal areas? So really, what does this mean and how do we inform management? Because we can't really go look at these numbers and say, well, we know that the coastal system varies a lot, but how much does it vary? And what does it mean for the biology of the critters there? So Dick Feely has done some really nice work as part of the Cal Coffee data. And basically what I want you to get out of this is that these are a bunch of different variables, temperature, aragonite saturation, pH, DIC, and PCO2. And what you see here is basically the ocean is really, really structured. It's beautiful. There's diversity along with the depth. But we're concerned with this really, really shallow area. What's going on there? There's not much data there, actually, but more and more data is coming out. So take a look at this map that you may be familiar with, which is the Monterey Bay Region. It's a bathymetric map. And right away, you see the canyon. There's some ridge mounds, really crazy diversity and structure, the canyons, all the little different watersheds, just like what you see on the land side. And again, you see the coastline, gap in knowledge. What's the structure there? And what's the different environmental variables doing there over time? Well, for the Monterey Bay Area, here's an example of one season, one upwelling season. We see that dissolved oxygen gets down to 50 micromolars, or about 1.5 to 2 milligrams per liter at 9 degrees Celsius. That's an upwelling event. During normal times, it should be saturated, about 350 to 400 micromoles. And it's about 12 to 13 degrees. This is just the Monterey region. Now, if you go further south, like Southern California, Baja California, which are also upwelling regions, this might be a different story. So, but we want to tell the story that's going on here for the abalone, especially in the red abalone. What about other variables? So pH, we're already seeing 7.6 during an upwelling event. So if we're thinking that global average is 8.0 and our experiments are aimed at minus 0.4 from here, well, the abalone are already experiencing these events. So we have to consider locally and regionally how we design our experiments and what the animals are exposed to. So again, 9 degrees Celsius generally, with the deeper, colder water coming up onto the shelf into the shallow water zone, and then 8.1, 8.0 at 12 to 13 degrees. So our organism of interest is the abalone. It's a really interesting critter. But raise of hands, how many people know what an abalone is? Okay, not everybody, but most people, which is awesome. You may have seen postcards like this in different shops. Here's an example of a postcard from Santa Barbara, 1950s around. Look at this shell mound up to the rafters of a house. You probably won't see something like this anymore in California. Now go back a little further in time. Anybody recognize this guy? Yeah, he's the quote-unquote abalone king of Monterey. The interesting thing about Monterey is it was really the heart of the abalone industry back in the heyday. All of the abalone from different parts of California, Baja, came here, got processed, and got sent elsewhere. The other thing this guy has done is basically popularized the abalone recipe for the Western world. He's a German restaurateur. He made a schnitzel recipe out of abalone. So bread it, butter it, cook it up, and all good. The other thing this picture tells me, look at the size of the abalone shells. Can we even find these guys right now along this coast? This is probably from Pacific Grove. When do you think this picture was taken? '89. Well, the car is obviously a clue, but this picture was taken in September of 2016. This is down in Baja, about central Baja. This is green abalone on the left, pink abalone on the right, and together they represent about 40 tons. This is from one seasonal catch from one fishing cooperative down there. So in the 80s and 90s, you guys may have heard about the abalone withering foot disease syndrome. That's a big concern. We think that El Nino events possibly cause a greater prevalence of that disease. It's still a big concern for the black abalone in particular. We know that 2 of the abalone species are on the endangered species list. So this is still going on in many parts of the abalone distribution. This is a great green abalone. On the both of them are green abalone. Here's a healthy one, and here's a sick one. But there are other diseases that may affect the fitness or the condition of the abalone. So if we look at the life cycle, just at the abalone, not species interactions or predation, they're basically considered gonochoric, male and females, although there has been reports that they may be hermaphroditic as well. They release their eggs into the water column and their sperm, and fertilization occurs in the water. Then it goes through a larval developmental stage, about 7 to 8 days for red abalone. Then at some point, they need to find a settlement queue, at which point they go through metamorphosis and become a baby abalone and go through a maturation process, which can take anywhere from 4 to 7 years, depending on the species, and then go through that whole life cycle again. But we look at it and say, what is going on at every one of these stages with all these environmental factors? In particular, we're interested in temperature, pH, and dissolved oxygen because those are some major environmental drivers. But there are more environmental factors, of course, but these are what is usually addressed in literature. So we began about 1.5 years ago on fertilization. What happens when you expose sperm and eggs to different multiple stressors or multiple drivers at the same time? So we're looking at dissolved oxygen, pH, and temperature for these experiments. And how do we do this? Well, first, we know biological factors matter. There's a whole host of literature, especially in the aquaculture literature, that there's optimal fertilization based on sperm concentration or egg and sperm ratios. We also know gamete age matters. Here you have egg age, and fertilization success goes really down as the gametes age. But again, we need to put this in context of the local regional area. Upwelling for oxygen events, low oxygen events, and low pH events at low temperature that we've seen in nature. So how do we do this? We take abalone, farmed abalone from Davenport, get them to individually spawn, collect the gametes separately, pull them together, and in gas-tight glass syringes with known seawater chemistry, we add the gametes. Here you see the eggs being added, and then that'll be followed by the sperm. And we allow them to be exposed for a certain duration of time for the experiment at different temperatures or a constant temperature. And then we measure the seawater chemistry, pH, dissolved oxygen temperature for each treatment. And 4 hours later, we assess their fertilization success. And this is kind of the background of what happens during these types of experiments where you have to get the animals to spawn and put them into experimental conditions. And you have to coordinate every single person here. So our first experiment was, let's just take a sledgehammer to these guys. Let's give them really low pH and really low dissolved oxygen. And we see from relative to high pH and high dissolved oxygen that we're already seeing a drop in fertilization success. We did it for different exposures because we wanted to determine that, well, we know if they're briefly exposed to a seawater condition, that might not be enough to see an effect. So we wanted to be sure that we have a relatively stable parameter to work with. So right away, we see that pH and dissolved oxygen has an effect on fertilization rates. But now the question becomes, which is more dominant, or are they interacting? So we created 40 plus levels of pH coupled with DO or temperature and put them into these incubators and assess fertilization after 4 hours. Here you see the data from our first set of experiments where we exposed the gametes to pH ranging from 7.15 basically to 7.9 under 2 different oxygen conditions. And basically you see the trend is very linear. There's no apparent threshold. And that pH and has a really dominant effect, and dissolved oxygen levels are really negligible on fertilization success. The blue dots represent high dissolved oxygen, and the brown dots represent the low dissolved oxygen. But what about temperature? So we exposed the gametes in a different set of experiments to 3 different levels of temperature. We have 9 degrees represented by upwelling scenarios, along with the same pH gradient levels or range of pH levels. And then black dots represent the kind of the control ambient levels. And then the red dots represent 18 degrees. The 18 degrees represents about Enso conditions in the Monterey Bay region. 19 degrees is about as warm as it's going to get. In the future scenario, that might change, but it allows us to assess how conditions, when it gets really warm, might affect fertilization rates. So again, you see that fertilization is independent of temperature. It's decreasing with pH, and that response is linear without an apparent threshold. But interestingly, this time, temperature is really having an effect. As you go from 9 degrees, 13 to 18 degrees, temperature is becoming more dominant or mitigating the effects of pH, which is, for us, it's like, how do we understand this? We think temperature can increase or decrease fertilization rates, but there's an interaction with pH. So if you based all your thinking on just temperature alone or pH alone, then you wouldn't understand that interactive effect. So in summary, we know that right now, ocean acidification would be a major threat to fertilization. But right now, dissolved oxygen or low dissolved oxygen or hypoxic events are really negligible for fertilization. But if ocean warming co-occurred with low pH events, these upwelling events, that would have a mitigating effect on fertilization. But based on these data, we're already seeing that abalone life history, as a small portion of it, is really vulnerable to how the environment co-varies together depending on the factors that are there. So what are we doing now? This work, the fertilization work, took over a year to do. There's a lot of, as Peter might say, there's a lot of details to figure out. And Peter actually was awesome at giving us some guidelines to how to do the aquaculture process. But once you get that figured out, you can repeat the process and do it with possibly other species as well. So really, the ocean is not stagnant. It moves. Water is being upwelled. It shifts around along the coast. There's dynamic exposure to the life history of the abalone. So we're interested in, what about variability? What if we have a control that varies at an ambient level, and another one that shows upwelling, and another one that's maybe a future projection of that, that tells us, well, we don't just focus on constant conditions, but how things vary over time. We've already seen some work established by Dave Layton in the 70s with temperature. These are Southern California red abalone. And temperature definitely has an effect. There's a whole host of literature out there that temperature is really significant on the physiology and actually the chemistry of that reacts in different ways in different rates. So we're already seeing that, but what about, as we said before, what if things co-vary with these variables? So the amazing team at Embari, led by Jim Berry, Dale Graves, Chris Lavera, and Chad Kesey, who's missing in action with the picture, decided we're going to build an upwelling system that we're going to vary the exposure dynamically so that it reflects the tidal cycles and upwelling events that we are measuring along our coastlines. So using a relatively simple control tank with a cooler, with seawater coming in, controlled by a computer and software, they're trying to vary the seawater chemistry in terms of temperature, pH, and dissolved oxygen so that we can expose animals from larval development all the way up to adults and see how they respond over time. So here's an example. Here's a first prototype or second prototype of an experimental chamber. The larval development in those chambers are going really well. We have to be able to control if we put 10 larvae in the chambers, we want 10 larvae to survive and settle really well. If we can't establish that, then we have to keep going back at it. So here's some data from the upwelling system trial. You see that over several days, they can cycle dissolved oxygen from 10 milligrams per liter all the way down to almost 2 milligrams per liter over the space of a tidal cycle. And during that same time, you can vary the pH levels from 8.1 all the way down to 7.2, and we can maintain that over several day period. So what's the future looking like for our experiment? Well, we have our undergrads leading the way. Here's two interns from this last summer, RU interns. They're scheming a timeline of their experiments and saying, okay, at this day, I got to do this. And she's saying, okay, on this day, I got to do this. So everything is laid out, and they're actually doing some of the experiments that we're interested in, but we're not sure whether it would be good to further develop and devote our resources. So they're actually leading that way. So here's Tiffany Thisner. She looked at low pH effects on sperm motility because we saw fertilization rates decrease with lower pH. So maybe sperm motility is affected and causing that decrease in fertilization. So this, actually, using ImageJ software, she can actually manually track 10 sperm out of a sample and get their swim rates, distance traveled over a period of time. And what did she find? She found that with a 1-minute exposure to low pH, she found that there is no significant difference between high pH, represented by the blue dot, and low pH, represented by the red dot. But what was interesting was that as the gametes aged, there was a significant effect. So here, the biological factor, or something that represents the gamete age, is having more of an effect than pH. So we can't forget about that. So that's an interesting result. Then we had another student a couple years ago, or a year ago. She looked at single versus double stressors during larval development. So what she did was take one group of animals she exposed to low DO and high pH, and another to low DO and low pH, a dual stressor. So what she found was that the triangles represent the average of the response. The dots represent the individual samples that she measured. The blue line represents the control, the high DO and high pH. With the single stressor of low DO, she found the development to be lower. In this case, development defined by hatching success. The moment that a trochophore actually hatches from its embryonic envelope and starts its swimming stage. So, but under dual stressors of low DO and low pH, there is a significant signal. So there's an interesting story there as well. So I love this picture because these are two of our undergrads this year. This is sorry, her name just popped out of my head. But this is Tiffany, and this is Daryl. And this is Daryl's project, looking at different sizes of juvenile abalone and their response to changes in temperature and changes in pH. So what she did was do an offset of the incoming seawater for temperature and pH. So a warming offset and a lower pH offset from the incoming seawater, and expose juvenile abalone to about 6 weeks under those conditions. She hasn't shown us the final data yet, so that'll have to come another time. This kind of work really can't be done by individuals or a single lab. The work has to be done by interdisciplinary thinking. The physical oceanographers, the ecological modelers, and the experimental biologists coming together and saying, well, these are some of the interesting questions here. How do we take what we observe in nature and bring it into the lab and test some of those questions? So this research is really led by Professor Fiorenza Micheli, Jim Barry, and Giulio DeLeo. And they're also right in there with the experiment during the experiments, playing with the gametes, injecting. So it's great to see 3 PIs in there, and I get to say, oh, you have to do that. So, and again, there's an army of volunteers and people who helped on this project. As I said before, Peter Hain, I used to come over here a lot before the project even started and said, hey, how do you do all the culturing? How do you do the handling process? Then I have to adapt that into our experimental setting. Say, our experimental container is that big. So we have to measure the seawater chemistry, all the different variables. So it has to be adapted. And then people who led the way a long time ago, actually, Dan Morse, who was my advisor at UCSB, talked to him a lot. And then just the engineers, I can finish there, but the engineers at Embari who are so, what do you want to do? Okay, we can solve that. That attitude is really, really, really amazing. So with that, I'll wrap up and take any questions. I think I blazed through this talk. That normally takes me a little bit longer. Yeah.
So it's talk about the dynamic environment, especially in upwelling zones.
Yeah.
Additional work's been done with develop on the fact that they do so well during upwelling periods, which are obviously those really low pH time periods. Yeah. When you're looking at 0.4, let's say, right?
Yeah.
Talk about that drop. There's a tendency to want to say, well, let's put 0.4 on to 7.6 and look at 7.2. But that deep water is coming. It's an older water source, right? What's the likelihood that surface
I think you're getting at one of the most fundamental questions that's facing the experimental biologists and the way we think about climate change.
Awesome.
So, and it's really good to be clear on why we design our experiments the way we do. So, taking point that what you said, that the atmospheric gas absorption mostly happens at the surface of the water, but that water sinks through biological pump, through particular pump, through other processes of mixing. So it's not inconceivable that the water that is currently out there in the deeper water zone in several hundred meters, that's around 7.6 or slightly less, would actually decrease in pH because of that sink of carbon. So we think about it and go, well, in our context, we think that could our 7.6 during upwelling could decrease in the future. So, but if we're talking about an experiment where we say, well, the near coastal zone only experiences 8.0 and maybe a 0.1 deviation from that, which could happen in some areas, then it's appropriate to say, well, upwelling or different dynamics, yeah. But we really have to start thinking about how the water masses move and how it changes because of the addition of the CO2. We're taking it in context here as saying, well, we think that 7.6 is going to decrease. So, but to cover all the bases, we're going to test everything from 7.1 to 8.0. So it becomes a challenge because the experiments that are currently designed are very orthogonal in tradition. So you basically test 2 or 3 levels of each, maybe if you have 2 stressors involved. So how can you test all of that? What might be happening based on a local regional context? Because the tropics, for example, may not be experiencing that at all. But here, certainly, they're already experiencing it. So I think the language has to kind of be changed a little bit in terms of here's our animals, in what local adapted capacity are we talking about? Because the animals, even for red abalone, rufescens, the animals that are in Northern California up to Oregon are experiencing totally different environmental regimes than down to Baja. So red abalone have historically gone down to about central Baja California. Down there, temperatures range up to 26, 27 almost every year in some places. And the abalone are doing fine. So if we apply that thinking to Monterey abalone, we're talking apples and oranges. So we do, I think the language, even the reviewers that commented on that fertilization manuscript, they were saying, well, the future predictions is minus 0.4 from 8. We're like, no, then we've miscommunicated that. Local variability matters. That's what our main message here is trying to say, I think.
The reason I brought it up was because I'm familiar with the OTEC project out in Big Island, which has red abalone growing in constantly cold,
Right.
high CO2 waters. And so it gets rid of that dynamic aspect. So it's potentially a control.
Right.
We're looking at red abalone growing at constant versus the dynamic point. So I was trying to consider the deep source and how that would be at play there.
Yeah, I mean, I think of it as, there's some talk in some circles, like, have all the big things been discovered and explored? I'm like, we're just scratching the surface because co-varying factors matter, just in terms of the environmental conditions. And we're just now learning how to establish systems so that we can test some of those ideas. I know that abalone farm in Hawaii, and it's like they get their water from super deep to keep them cool. But then there's abalone that are raised in really warm temperatures as well. So it's this. I think the local adaptation is a really key thing that we need to understand. Yeah.
I was curious about the seasonality of the timing of spawning in the wild with the red abalone in relation to when the upwelling events happen, right? If they spawn when there's upwelling, that could be bad, but if they're spawning at a different season,
Yeah.
maybe it's not so bad. So I was just kind of curious what's the typical pattern, at least in our backyard, for when they spawn.
So actually, that's a really awesome question that I've neglected to ask the audience today. Some of you who know what abalone are. How many know when abalone spawn? Okay, Peter does. What time?
They can potentially spawn any month of the year. Most of them spawn during the early spring,
Right. But what time of the day?
nighttime.
Nighttime. Like a specific nighttime point in night? Midnight, 2 in the morning? Well, these are actually very appropriate questions because fertilization happens or spawning happens like that, right? It lasts for 10 minutes, depending on the population and how aggregated they are and that synchrony. I asked this question like all up and down the coast, commercial fishermen who made their living at collecting abalone. None of them could answer that question. What time of the day do abalone spawn? Because upwelling can happen with the tidal cycles. That's within a day. And within a day, that variability can go from 7.6 up to 8. So we actually don't know out in nature. There's seasonality of spawning that's been reported, but there is zero primary publication about exactly what time of the day they actually spawn. If you go into the coral world, that's a complete story, a different story. There's tons of literature about this species spawns at this time of the year at this time of the day, right? And that's pretty accurate. Abalone, we don't know. In the lab, we know, and there are some evidence from people who've been around abalone a long time, but a documented, repeatable observation has not been reported. So the question is open. So the way that we trigger abalone to spawn is to raise the pH and then really dramatically drop the pH. Along with that, we also raise the temperature about 4 degrees from start. So we've had 95% success rate with all the different iterations that we've had for causing both males and females to spawn, and females spawn later than males. So there's that game to be played. So they're cueing on what the seawater chemistry is doing. We know that. Dan Morse has established a lot of that work. But you know, can we say, based on our lab knowledge, that's what's happening in nature? There's a lot of factors, obviously, that goes into that. But I think that just looking at the life history, I think is super interesting. I tell all the students who work with us, I go, you're going to become a master of life history, and you're going to become a master of time. When do things happen? Because it's hard to talk about when things happen and the way it should if we don't have a good idea. So within all this is pages and pages of timestamps. So it's great to have people like Peter Hain to talk to and say, this is what I do. And we go, oh, okay, that's pretty cool. And just to put a time recipe to it. So I think one of the other things that we could do is starting to go out to the natural system and start documenting when things spawn, not just abalone, but many other critters that are maybe especially economically relevant, and say, well, this part of the year, this time of the day, this is really critical for the life history processes and the population parameters. So we might be a little bit more careful what's happening along the coastline. For example, light has been shown to disrupt coral spawning synchrony. Places around the world are now turning off the lights along the coast during that time of the year. So it's informing not only science, but the coastal management as well. So there's plenty to be learned about out there. But awesome question. Yeah.
Do these environmental variations affect the gamete quality in the broodstock? Are you
Yeah.
taking sperm and eggs out of spawning abalone
Yeah.
and say, okay, we're starting, here's 0.0, but
Yeah.
are they different abalone from different places
Yeah.
affecting the gamete quality?
So that's a very good question. There's two questions buried in there. One is, if I understand it correctly, one is, does the conditioning time during the brooding time affect gamete quality? And the other might be, if you have broodstock from different areas, would they respond differentially? Those are very good questions that we're very interested in doing those kinds of experiments. Laura Rogers Bennett, I don't know, 10, 15 years ago, did some experiments with broodstock and temperature, and she showed that 18 degrees had a negative effect on gamete quality for red abalone species. Now, here, which is really interesting, right? We've tested 18 degrees for fertilization, and then that increased the fertilization rate, but it decreases the broodstock quality. So our approach still has to be a complete holistic approach, saying maybe the net outcome of all the different stages is what we also need to consider in order to kind of predict the demographics and population changes over time. So it's not enough to just look at fertilization and say, this is obviously what pH, temperature, and DO is doing to everything. So that's why we're looking at different stages of the life history. So I don't know yet, but temperature certainly has an effect on that quality. Yeah. All right. You survived. Thank you.